Hey, Bio2 students, Mr. Yance again. Hope everything is going well. Let's see if we can go ahead and finish up the notes so you can get your foldable done and you guys can take your test Monday, okay? So we're going to talk a bit about cell junctions. Not all of these, again, are in your foldable. Some of these are just simply questions I'll ask on the Part 2 test on next Wednesday. When you're dealing with plants, they have these cell walls, and these cell walls are super thick. And since they are super thick, we have a problem. We have to get things going through there. CO2's got to come in, oxygen's got to go out in a plant, and the only way to do that is to poke holes in that cell wall so that things can flow through it. And we have a fancy name for that. It's called plasdesmatas. You can see these little holes. This is a cell wall, and we just poke holes in them every so often, and that allows things to flow through that plant cell. Okay, nothing more than that. Just pretty simple, straightforward holes in a cell wall. In animals, we also have some unique structures, and there's three of them I want to kind of focus in on. We have tight junctions, which is where two cell membranes will basically glue together. And then there's what we call desmosomes. Let me give you a very simple illustration on that. Just give me a second. I'm going to pause. I'll come back. Okay, so let me give you a pretty simple illustration. So remember back in kindergarten, we used to glue, and we still glue, paper together, right? And there's a couple different ways we can do that. We can take dots of glue and spread it around and glue it. Not quite as strong, but it works. Or I could take a big bead of glue and just pour it all over the sheets of paper. That's a stronger bond, right? So that's a tight junction. Tight junction would be a big bead of glue fusing cells together, tissues together. Desmosomes are the little dots of glues you see every so often, all right? You can see the difference here. This would be a desmosome. We just every so often we just put a little dot of glue, whereas a tight junction is going to be all the way down the length of those membranes. Okay, they do the same thing. They're just basically holding cells together, tissues together, but we do it two ways. We either use a dot of glue or we use a tight junction. And I talk a lot about that in my NAMI class too. So if you take my NAMI class, you may hear me say that too. And then we have these gap junctions. You can see these. Very similar to plasdesmata, except it's in an animal cell. And that's basically where we put little channels through like a tight junction where I got it sealed. I got to put a channel or a cut or a hole through there so things can flow between the cells and between the tissues. So it would be the animal version of the plasdesmata, which is the plant version. All right, so that's pretty much what they do. Okay, pretty simple. And then we have some things I kind of want to put in your foldable. Okay, let's talk a little bit about cell walls. Obviously, not in an animal cell, simply only in a plant cell. They protect, shape, and regulate. Okay. Now, I'm going to go in a little more detail than maybe I did in Bio 1, so we just talked about cell walls and left it at that. But in a cell wall, you have different layers. Now, remember that plant cells and animal cells both have plasma membranes. I think sometimes we get that misconception that plants have cell walls, we have plasma membranes. We both have plasma or cell membranes, the outer border of the cell. They just have this added stuff that's non-living on the outside of that. So a primary cell wall is laid down first, and then they put this thing called a middle lamella, which holds the cells together. It's actually a polysaccharide. It's called pectin. We use that in jellies and jams to make a gel. And then some trees especially will put a secondary cell wall, which is wood, bark, and that type of thing. You think about how big giant sequoias, giant redwoods, big oak trees in our woods. These secondary cell walls can be extremely thick and sturdy, and they have to be. If a tree gets to be 30, 40 feet tall, you better have some durability there. And you can see here's the primary cell wall. The pectin's in the middle, and then the secondary cell wall is added to the outside. All right, and that could be several layers thick as well. Okay, so this is this on the out. The plant cells on the inside, and we just add all this stuff on the outside. So plant cells don't have a luxury of having a skeletal system like you and I have. So how they get around that is by using a cell wall. That's what gives them support and protection and allows them to grow to the great big massive structures that some trees are, right? Okay, so that's that. Okay, chloroplast, another one only in plant cells. Chloroplasts contain chlorophyll. I'll go more into detail with the different types of chlorophyll A and B, when we get into photosynthesis chapter. A little confusing with chloroplast because there's three parts to a chloroplast, and this obviously is where photosynthesis is going to take place in a plant, the green part of a plant. Think of poker chips. So we have these poker chips that, let me show you a picture, that are called thylakoids. So where this gets a little bit confusing is a thylakoid is each of these individual green discs. Okay, the whole thing, a whole stack of them, is called granum. So if we're using the poker chip example, an individual poker chip would be a thylakoid, and I stack 10 of them on top of each other. That would be a granum or grana. Okay, and then the solution around the outside of all these granas is called the stroma. And I'll talk a lot about this in photosynthesis. We have something called the light dependent reaction, the light independent reaction. The light dependent occurs in the grana. The light independent occurs in the stroma. So they work together, and by using the combination of both of them, then that's how we carry out photosynthesis in a plant. All right. Not going to go in more detail. Just wanted to get you the basics so you understand the structure. We will get to function and go in a whole lot more detail with that in the future. All right. Let me show you the last couple things here, and I apologize. My students today told me, Mr. Yance, you didn't put these last three things in the notes. You're just going to have to write these in. So I apologize. For whatever reason, my notes didn't include these last little topics. All right. Still want you to know them. Still want to put them in a foldable. So we're still going to go over them. So I apologize. That's on me. So these last three, let's kind of wrap things up a little bit. Nuclear envelope, really simple. That's just a membrane around the nucleus. It maintains the shape of the nucleus and helps protect the nucleus from damage. Really simple. That's just this purple that goes around the outside of a nucleus. And if you look at the tiny little bitty holes there, that's going to be your nuclear pores. Okay. So, I didn't go where I wanted to. So that's basically the only difference between those two. Not really complicated. Not going to spend a ton of time on that. Oops, you know what? I just noticed something. My fault. I skipped one. You're probably wondering, where in the world is Mr. Yance at? Let's go back. Let's backtrack. We'll come back to nuclear envelope. My fault. So about cilia and flagella. I don't know if those are in your notes or not, but cilia and flagella are just simply cilia are hair-like structures. Flagella is more like a tail. Both are involved in locomotion. Both are involved with movement. I will post these notes online today. There's a couple good videos that show how these are different. The biggest difference is cilia just beat back and forth and produce a wave. A flagella is more like a whip-like tail, like sperm have on them. Cilia you find in your lungs and respiratory system. They do the same thing. They're both involved with movement. It's just a matter of how they do it. So is it a wave-like structure and a beating back and forth like this one, or does it have a whip-like type of design? So watch those two videos and you'll see the difference. Okay. Oops. Didn't want to play that. So you can watch that on your own time. All right. It's kind of funny to watch. Okay, yeah, that one's going to start too. Shoot. So let me see if I can get us out of this slide. All right, let's go back to what I skipped ahead to. Nuclear envelopes, again, just that membrane, and then the nuclear pore just allows many doorways. It allows things to flow through the nucleus. Okay, and the last thing, and then we'll be done with notes, is the cytoskeleton. These are just protein filaments that help maintain the shape and help the cell with movement. Okay, and again, it may seem like I kind of rush through these, but I just kind of want you guys to a little review. We went over these in Bio 1 a lot. We forget stuff though. I just kind of wanted you to get back in your brain. So the first part of the test is this. You're going to get your foldable out. I'm going to ask you a couple essays about these. And then the other part of the test will be matching multiple choice, that type of thing like we normally take. All right. The reason I'm not spending a ton of time on any of this stuff is because, again, we're going to have chapters on them later. All right. Hopefully that makes sense. Hopefully you get your notes all done, your foldable done. Do that over the weekend. And then next week we will get the 2 tests in. All right. Hey, have a good one. If you got questions, we'll have a Google Meet tomorrow anyway. If you have any other questions, shoot me an email. All right. Hey, we'll see you. Bye.